Assalamualaikum. Today we will presenting through the entire process of DNA analysis, starting from how we preserve our biological samples to extracting DNA, amplifying it using PCR, and finally visualizing the DNA using gel electrophoresis. Here's a quick overview of what we will cover today. The DNA workflow has four main stages. The first stage is sample preservation. Secondly, the DNA extraction. Then DNA amplification using PCR, and finally visualization through gel electrophoresis. Each step plays a crucial role in ensuring whether we can get clean and high quality DNA result. The first step is preserving your biological samples to prevent DNA degradation. Common methods include storing tissue in ethanol, usually 70% to 100% concentration, or using silica gel for drying. Cold storage is also effective, especially at -20 degrees or -80 degrees Celsius. Most importantly, always label the samples clearly with species name, collection date, and location. This because good labeling ensures proper data management for future references. For the second step, which is DNA extraction, once our samples are preserved, we extract the DNA. This involves breaking open the cells using a buffer like CTAB or STS, removing proteins with enzyme or phenylchloroform, and isolating the DNA with alcohol like ethanol or isopropanol. The final product is usually a small pellet of DNA, which we resuspend in a buffer for storage or analysis. To perform DNA extraction, we need some essential lab tools: mortars and pestle or homogenizer for tissue grinding, a centrifuge to separate DNA, micropipettes for accurate liquid handling, and a water bath or heat block for incubation step. And of course, we also need extraction buffer and chemicals to carry out the reaction.
Hello. So, in step 3, DNA application PCR. Now that we got our DNA, we amplify the target region using PCR, which is the polymerase chain reactions. This technique makes millions of copies of specific DNA sequence. We need a few ingredients: template DNA, primer, nucleotides, tag polymerase, and buffer. The process runs through the repeated cycle of heating and cooling, deterioration at around 94 Celsius, annealing at about 50 to 65 Celsius, as and extension at 72 Celsius. Typically, we do 25 to 35. So, in step 4, gel electrophoresis, to see whether the PCR works, we use the gel electrophoresis. First, we prepare the agarose gel and load our DNA sample into well. An electric current is passed through the gel, and because the DNA is negatively charged, it moves toward the positive electron. Smaller fragments move faster, so we can separate the DNA by size. We stain the gel with a dye like ethidium bromide or SYBR, safe to visual the DNA under UV lights. In interpreting the gel, here what you will typically see on the gel. You will see the DNA ladder as a size reference. Bright band shows successful implications. If you see no band, the PCR might have failed. Smearing could mean the DNA was degraded and too much template was used. Always compare your sample length to the ladder to estimate fragment size. Troubleshoot, Troubleshooting is a part of the process. If there are no bends, check if your primer are correct. And if your DNA was pure and intact, multiple band messages, not specific amplifications, possibly due to incorrect aniline temperature. Smearing usually means degraded DNA or too much DNA was used. Always include a positive control to verify the PCR is working.